and Intervention Technologies Research Unit at the U.S. Department of Agriculture's Eastern Regional Research Center, holds one patent with over 150 published peer-reviewed articles, critical reviews, and book chapters. Thank you so much for joining us, Dr. Niemira, and welcome.
Thank you very much. Thank you. I'd like to thank the organizers of the Oman Food Safety Conference for inviting me to share with you some of the technologies that we're working on in the USDA at our Eastern Regional Research Center. I'll go ahead and share the screen. Right. So just to give you a little bit of context, within the United States Department of Agriculture, we have our own research arm, an entire agency devoted to agricultural research. And we have research projects all across our country and really all around the world, covering everything from water safety, worker hygiene, pathogen residence in crops, crop conversion efficiency, breeding, lots of different kinds of applications. My own research unit, the project that I head, and the team of scientists that I'm responsible for, is the Food Safety and Intervention Technologies Research Unit. And what we do is to develop technologies to apply to foods, a wide array of commodities in a post-harvest setting to make the food as safe and as high quality as it could possibly be. We work on a lot of different kinds of technologies. I'm not going to talk about all of them, but we cover a lot of different areas that might be applicable for all sorts of different commodities, ranging from meat, poultry, eggs, fresh produce, seafood, shellfish, finfish, powdered foods, ingredients, and other kinds of things. What I'm going to be talking about, just sort of focus in on just a few of these technologies, are radio frequency energy processes, pulsed light, cold plasma, antimicrobial packaging, and active packaging. So first, let's talk about radio frequency egg pasteurization. Now, as you heard from the previous speaker, there's a great deal of effort that's put in to making sure that you have certified feed and good growing conditions to make the eggs as safe as possible. And as other speakers said, there's a lot of effort working with the workers and the training to make sure that everybody is on board in terms of what it means to have a good food safety culture. Despite all of this, there is still the possibility of having salmonella on eggs. In the United States and typically around the world, you have 1% or maybe a little bit less than that, or you have some salmonella that does get into the eggs. So you can certainly do testing, you can do holding and doing recalling on those. But we wanted to develop a technology that would allow you to be certain that your eggs are salmonella free. And to do that, we partnered with the Plasma Physics Institute at Princeton University, not very far from our center actually, to develop an advanced inside the shell egg pasteurization system. Now, there are pasteurization systems out there that use a warm water bath to allow heat to penetrate inside the egg to slowly inactivate the salmonella. It's a little bit slow, it's a little bit expensive. We wanted to do something very different. And we developed this technology that would allow us to pulse radio frequency energy inside the egg. So it's essentially heating the egg from the inside out in a very controlled sort of way. When the electricity comes in, it's differentially absorbed by the yolk because of its lipid content, as opposed to the albumin because of the protein and water content. And effectively what that does, it allows precision thermal inactivation exactly at the interface between the yolk and the white, which is where the salmonella is laid down as the chicken is formulating the egg. And what that does is it allows for very rapid inactivation of the salmonella. So you can have a 99.999% reduction of the salmonella, essentially salmonella-free eggs, while still retaining all of the performance characteristics of the egg. Typically, when you use the thermal bath treatment, you get a little bit of hazing of the yolk or hazing of the white. It doesn't perform as well in applications. But with this new technology, we have eggs that look, act, and perform from a functional standpoint almost identical to raw, untreated eggs. So with this, you can use these eggs in cooking and ingredients and for home use for people who are especially concerned with having these salmonella-free eggs. Right, so this is just one of the technologies that we're working on. Other areas that we're investigating are active packaging and pulse light. There are different kinds of ways that you can introduce antimicrobial compounds onto foods, and one of the things that's gained more interest of late is chlorine dioxide, gas phase chlorine dioxide. It's very reactive. It works very well. It's pretty clean, but in order to get it onto the food, you have to obviously apply it. Well, once that food is inside the package, it's very difficult to do any other kinds of treatments. Your options are pretty limited. Well, this technology actually involves taking the chemical precursors of the chlorine dioxide, sandwiching them between an inner layer, with an inner layer between them. All right, so they don't do anything. They sit there. They're totally inactive until you push the button. Okay, and you activate that. Activate the layer, and the two precursors start to mix with each other. All right, that's when the chemical reaction takes place and releases the chlorine dioxide into the package. So you can have the chlorine dioxide generated inside the package. So then you have post-packaging control of the pathogens on different kinds of products. Another activity that we're very heavily involved in is pulse light. So this is one, again, inside the package, where you can have high intensity light that shines through the packaging to inactivate E. coli or other kinds of pathogens. The longer the treatment you have, the better control you're going to get. So this is a treatment where we use the pulse light for E. coli contamination on romaine lettuce. The light was pulsed onto the packaging, and we had, better than 2 log, better than 3 log reductions based on just a very short treatment of about a minute. Okay, so total background microflora was also reduced by these treatments as well. So you had reduction in spoilage and other kinds of damage. So you had really good control of packaging, pathogens post-packaging. Right, it's pretty important. Last technology that I want to talk about, I'll go into this a little bit more detail because it's relatively new, is cold plasma. Now, cold plasma is, as I said, it's a relatively new technology. And what this technology does is it uses energy, typically high voltage electricity, to actually tear apart the atoms that make up a feed gas. Air is very common because it's very cheap, but you can also use things like a defined gas, like helium or argon, specific oxygen-nitrogen mixes. And what you get when you do that is all of the gas atoms are broken apart. So you get free electrons, you get ions, you get radical species, all of this very reactive chemical mix, which then will interact with the pathogens on the surface of whatever food you're treating to cause DNA damage, to cause membrane rupture, to inactivate those pathogens. Ultimately, what happens after that is you get a self-quenching plasma. What that means is that these reactive species, if they don't interact with the pathogen, they will react with each other, and they'll just return to oxygen or to nitrogen. All right. There's a whole mix of different kinds of compounds that are produced in this process. Sometimes the energy goes into the atom, it kicks off a high energy electron. And if that high energy electron doesn't interact with the food, it will rejoin the atom that it came from and then give off ultraviolet light. Sometimes you have O2, oxygen, which cracks into monoatomic oxygen. It's extremely reactive, but then that might react with itself to make O3, ozone, also very reactive as an antimicrobial. So you get a whole different mix of antimicrobial processes arising from this plasma creation system. It is relatively new. It's in some cases it's adapted from existing applications for processing of inks and paper and other kinds of non-food applications. Regulatory status of the technology is still being developed, so the laws that govern how it's used are still being formulated. This is just an example of what it looks like. Gas comes in, it's torn apart, and then you get a plasma that you can actually use to inactivate materials. You have a lot of different kinds of ways to make plasma, and what you're treating and how you're treating it has a big effect on how the plasma interacts with the surface, how much energy is captured, how effective it's going to be, whether you have a slow, gentle process, which might take a little bit longer, or a very rapid, aggressive process, which might be a little bit quicker. This is an example of one treatment that we did inactivating norovirus surrogates on berries. Here, blueberries were used as a model for a small, soft, fragile fruit. What we found is that the plasma was very effective at inactivating both of these norovirus surrogates, two-lane virus and murine norovirus. The longer you treated with the plasma, the better the inactivation. And this was true irrespective of whether or not there were engineering controls to bring the temperature of the plasma down, clearly below what you would see as a thermal inactivation. So we know that this is not just a matter of heat, okay, as an inactivation function. This is actually a chemical process of the plasma that's causing the disruption of the viruses. So this is another treatment that we did. We inoculated salmonella on the surfaces of oranges. Typically, it's a little bit easier to inactivate salmonella when it's on the peel or on the outside of the fruit, as opposed to when it's down deep inside the stem scar or inside the blossom end. Okay, it's much harder to inactivate those. So when we did this treatment, we did it at a couple of different distances. Very close, which would be fairly aggressive kind of a treatment. A little bit farther away, which is a little bit gentler. And we found that no matter which treatment formulation we used, we got very good inactivation. Even down deep inside the stem scars where ordinary chemical treatments won't be able to reach, as well as inside the blossom, we were getting in excess of 3 and up to 4 logs of reduction of these. And that was true whether or not we had the orange very close to the plasma head or farther away. So the longer the treatment time, the greater the reductions. So what's the big advantage over conventional chemical sanitizers? Right? I mean, why go through the trouble doing cold plasma as opposed to sodium hypochlorite or some other kind of chemical wash? There's no chemicals that are going into this. No exogenously applied sanitizer compounds. The only inputs to this cold plasma system are air and electricity. So this means that you have a much wider application for specialty applications where people might not want to have chemical sanitizers. The other thing is that when the compounds, when the plasma resolves and all those compounds react with each other to make the quenched plasma, there's no chemical residues. So you're not spraying anything on, you're not having to wash anything off afterwards. Another advantage is this is entirely a dry process. So if you're doing other kinds of products, such as, say, cilantro, herbs, leafy greens, if you put on a wet treatment, then you've got water resident on the leaves. That can then lead to the growth of spoilage organisms. So in a dry process, you don't have that, and this preserves the quality of the food for a lot longer. This is another example where here this time we were looking at different, not so much bacterial pathogens, but a parasite, Cryptosporidium parvum. So we put oocytes onto the cilantro. We treated them in plasma, as you see, for various periods of time, and we got better than 2 log reductions. Now, at the longest treatment time, you did start to see a little bit of darkening, a little bit of change. So there's going to be some kind of an optimum treatment level where you get the inactivation of these very difficult to kill organisms and the quality, right? So this is process optimization, but you've got this inactivation you can then build on these very sensitive, very difficult to treat commodities and very hard to kill pathogen targets. Another example of hard to kill things is biofilms. Now, typically we think of bacteria as growing individually, separately, but bacteria actually spend a lot of their time in dense, matted communities of organisms called biofilms. They're very sticky, very hard to kill. Sometimes you have to give 10, 100, 1,000 times as intense treatment if you're using chemical sanitizers as if to treat biofilms, as opposed to when you're treating individually planktonically living pathogens. When we applied this biofilm apply the cold plasma to biofilms of E. coli or salmonella, even very long-growing, very mature biofilms, very difficult to kill, we got very good results with very short treatment times. The thinking is that if material is going down a conveyor belt, you're not going to have a lot of time to treat it. So how much kill can you get in an industrially relevant time frame? 5 seconds. 10 seconds, 15 seconds. And what we found that with E. coli and with salmonella, E. coli, we got up to 3 logs reduction, so 99.9% reduction. Salmonella, which is always a little bit harder to kill, we got better than 2 log reductions, again, with these very, very rapid, very short treatment times. And just the thermal analysis that we did of this, looking at different pulse frequencies for the plasma production, showed that, yeah, this is not heat. It's not just that we're heating up the biofilm, that's what's killing it. These are still well below the point where you would see thermal inactivation. So this is truly a non-thermal process that is using the chemical species related to the plasma to do the inactivation. The last thing I want to talk about is combining cold plasma with other processes. This is an inactivation of Listeria monocytogenes on Granny Smith apples. A niacin-based sanitizer was able to give us 3.5 logs, so 99.98% reduction with a 60-minute residence time. You leave it on, let it work, it'll inactivate. If you treat that listeria inoculated apple for 40 seconds with plasma, then apply the sanitizer, you get the same 99.98% reduction. But now instead of it taking 60 minutes of residence time, it only takes 3 minutes. The plasma is causing a sublethal injury to the bacteria, which makes them more susceptible to subsequent treatment. This is pretty exciting because it allows plans for combining cold plasma with other sanitizers to make each of them work better together to leverage the strengths of what they can bring to it. So, just quick conclusion, there's a lot of things that we have been working on. A lot of things are, very mature and they're, coming near to market. Everything that we do, we always work with partners in universities and in industry to transfer these technologies out to where they can help consumers, right? That's what we do. We are a government agency and a taxpayer-funded organization, so we do interact with people. We want to make sure that people are able to use these technologies for foods and for companies to leverage them to solve existing problems and to pursue new opportunities. There's lots of different areas for future research that we're currently engaged in and we're going forward. But I do want to say, again, thank the organizers for their, for the kind invitation to speak to you today. If you have any questions, I'd be happy to talk to you. My contact information is here, and you can follow up, as you like. Thank you very much.
Thank you.